Hello everybody, and welcome to the second in a series of electronic seminars funded by Defra and hosted by LGC. This second e-seminar is based on a Defra Knowledge Transfer event entitled DNA Extraction Approaches to Support Food Labelling Enforcement, and which was organised and hosted by LGC on the 5th of March 2014 and originally presented by Malcolm Burns, Gavin Nixon and Tim Wilkes. My name is Tim Wilkes and I will be the presenter for this current e-seminar. Before we start, I'd like to go through some important points of information pertaining to a Defra Knowledge Transfer event that was delivered at LGC on the 5th of March 2014. Although every effort was made to ensure that the information was correct at the time of delivery of the original Knowledge Transfer event, LGC and Defra assume no liability on the accuracy of the information. LGC and Defra retain all copyright associated with the following e-seminar, including the use of audio recordings and visual images. All rights are reserved. You may not distribute or commercially exploit the content, you may not transmit the content or store it in any other website or other form of electronic retrieval system without the express written permission from LGC and Defra. We will now go through a series of introductory slides in order to provide some background information regarding the current e-seminar. As an overview of this section of the e-seminar, we will provide a brief description regarding the background to this e-seminar, put the process of DNA extraction into the context with respect to DNA molecular analysis workflows, and state the main aims of the e-seminar itself. Obtaining high-quality intact DNA is the first critical step in many fundamental molecular biology applications, for example PCR or DNA sequencing. It is also an important aspect of many of the published Defra and FSA protocols that use molecular biology approaches for food authenticity testing. The extracted DNA must be of sufficient yield and quality to be amenable to use in downstream molecular biology applications. But isolating intact total DNA from tissue samples can be a challenge and is dependent upon a range of factors. There's therefore a requirement to provide programmatic guidance and share expertise on how to select DNA extraction methods that are appropriate for use with representative samples. The flow diagram shown here illustrates the different steps that comprise a typical DNA-based analysis, in this example PCR-based analysis. As can be clearly seen from this figure, effective DNA extraction underpins all of the remaining procedures and is therefore critical to a successful outcome. If we now consider just the molecular biology analysis component, we can see that the DNA extraction procedure is comprised of a number of different steps. These include the actual sample preparation, which can include mixing, homogenisation and subsampling, all of which ensure the accurate representation of the sample material. This is followed by DNA extraction, which can be a general procedure or matrix specific, and which may be derived from in-house methodologies or with use of a commercial kit. And finally, some form of sample quality control and quantification is performed. The aim of this e-seminar is to provide access to the information first presented at the original Knowledge Transfer event that was held at LGC on the 5th of March 2014. This will focus on the dissemination of best practice for the extraction of high quality DNA from a range of sample types in order to apply the downstream molecular biology methods from FSA and Defra protocols with assurance. We will include a review of current Defra and FSA food authenticity protocols. An introduction to the principles of DNA extraction will be presented, and these will include the implications of using different food matrices, sample preparation including homogenisation and representative sampling, the principles of DNA extraction approaches, and the characterisation of the extracted DNA through DNA quantitation or yield determination, and DNA quality control, which assesses DNA purity, the presence of interference, and the degree of degradation or integrity. Continuing with the aims of the seminar from the previous slide, guidance will be provided on the extraction of DNA from a range of different food matrices. This will include an overview of the methodologies employed by both the UK and other EU official control laboratories. Current common approaches for DNA extraction will be discussed and examples of kits and techniques presented. There will also be a short section on DNA extraction troubleshooting. Guidance on sample characterisation of the extracted DNA will be provided, including recommendations on how to perform this and on how to interpret the results. There will then follow a section on the presence of matrix interference, including a discussion on how best to test for this and on how to interpret any test results. Finally, a short section comprised of a number of decision trees will be presented and which describe one possible route of approaching the issue of DNA extraction through a series of logical steps. These trees are for reference only and do not constitute an LGC Defra authoritative document and do not have any legal status in a court of law. We will now go through a presentation which will describe the general principles underpinning the process of DNA extraction. As an overview to this presentation, we will talk a little about different food matrices, sampling preparation, including homogenisation and representative sampling, the principles of DNA extraction approaches, and characterisation of extracted DNA, including DNA quantitation and DNA quality control. We will now describe the impact that different food ingredients can have on the process of DNA extraction. On introducing the topic of DNA extraction from food samples, it is important to recognise that food matrices are complex. Foods both native and processed are comprised of different categories of compound which may impact on the method required to extract DNA from them. Fundamental components include fats and oils, proteins, carbohydrates, salts and water. And exotic components which may be found include polysaccharides, polyphenolics and various synthetic additives. It's the combination of these compounds that will define the DNA extraction method that is most applicable for use. The complex nature of the composition of a food is illustrated in this slide by considering the ingredients found in a pizza. The dough base is comprised of a mixture of polysaccharides and proteins, whereas the tomato paste with which it is topped tends to be high in salts and polyphenolics. Even the cheese and the salami topping is comprised of a mixture of fats, proteins and salt. The impact that food ingredients can have on many downstream molecular biology processes is illustrated here. Carbohydrates and polysaccharides, which principally originate from plant materials, prevent the solubilization of DNA and inhibit both PCR reactions and restriction digests, whereas polyphenolics, which are primarily products of plant secondary metabolism, can inhibit PCR and also restriction digests. Synthetic additives, which include compounds such as food dyes, preservatives and sweeteners, also inhibit PCR and restriction digests. In the following section, we will consider various aspects of the sampling process and how they can impact on the typical molecular biology analysis. Sampling of food matrices is inherently problematic due to its heterogeneous composition and the presence of components such as carbohydrates, lipids and proteins. Effective sample preparation strategies are central to representative DNA extraction. Unfortunately, there is no universal sampling approach available, and selection of an appropriate method is on a case-by-case basis. Key to any sampling approach, however, is the assurance that accurate representative sampling is achieved. The key aspects that govern this are homogenisation and representative sampling. Homogenisation ensures that a sample's composition is the same throughout and facilitates the taking of a representative subsample. The method employed for this is generally matrix specific, and two examples for achieving homogenisation are illustrated here. The first being the conventional mortar and pestle, which requires the physical grinding of a sample, and a more modern bead mill homogeniser, in which samples are dissociated and homogenised through the reciprocal agitation of a sample in the presence of hard inert beads. Once homogenisation of a laboratory sample has been achieved, the process of subsampling can be implemented. This involves the taking of small subsamples of the original homogenised laboratory sample in order to guarantee an accurate evaluation with no positional bias. In an ideal world, the more subsamples taken the better, as this can reduce the limit of the level of detection achievable. However, the number of subsamples that can be realistically taken is frequently limited by cost and practicality. One important overriding factor for any sampling procedure is that it must avoid the chances of cross-contamination. An example of a typical sample procedure is illustrated here, and which has been taken from the official EURL guidance document regarding the detection of unauthorised Chinese GMO rice. In the next few slides, we will talk a little about the principles underlying the process of nucleic acid purification. As an overview to this section, we will look at the role of DNA extraction in the isolation, concentration and purification of DNA. We will look at some of the general approaches used to extract DNA, both in-house and kit-based. We will then look at the procedure in a little more detail, and then look at some of the example approaches, including both solution-based and kit-based. The objective of any DNA extraction procedure is to isolate, concentrate and purify DNA from a sample. This DNA needs to be representative of the sample itself, of high quality, with the removal of downstream process inhibitors. It needs to be repeatable and reproducible, ideally rapid and of low cost. However, the method selection will depend on the sample matrix and the downstream processing requirements, for example, PCR analysis or DNA sequencing. The types of DNA extraction procedure can be broadly categorised on whether they are solution-based, such as those that employ Phenol-chloroform, CTAB or SDS, those that are a combination of solution and solid phase, for example, CTAB with the additional use of a cleanup column, or lastly, those methods which are primarily based on a solid phase extraction, and where the solid phase could be a column, a resin, or DNA binding beads or particles. If we were to consider DNA extraction in terms of a process, then the following diagram illustrates the workflow involved. In step 1, we have sample disruption. This is then followed in step 2 by the release of the DNA into solution and removal of cellular components. And finally, in step 3, the DNA is purified and concentrated, either as a pellet or as a liquid elutant, depending on the method employed. The first step in any general DNA extraction procedure is the disruption of the extracellular components to release the DNA into solution. To achieve this, we can use detergents to disrupt both lipid and protein-based structures, and these can include reagents such as CTAB or SDS. We also frequently use enzymes such as proteinase K and alpha amylase to solubilize cellular structures and inactivate nucleases. Quite often we employ chaotropic salts which help denature proteins and inactivate nucleases, and typical examples of these compounds include guanidine hydrochloride and urea. And finally, all of this is maintained in a buffer which helps maintain a stable pH and inactivate nucleases. Typical examples of buffer components include trisma and EDTA. Following the release of DNA into solution, we need to remove all of the residual cellular components. Removal of membrane lipids is achieved by the addition of detergents or surfactants, by organic extraction, or indirectly by the use of ion exchange matrices. Removal of proteins is performed by the addition of a protease, such as proteinase K, and this is an optional step that is frequently performed. It can also be achieved by the use of organic extraction, with the use of compounds such as chloroform, or by the use of ion exchange matrices. Removal of polysaccharides is achieved with the use of some detergents in conjunction with selective precipitation, and removal of RNA is performed by the addition of an RNase. Proceeding to step 3, the solubilized purified DNA needs to be isolated. In the case of solution-based DNA extractions, this is normally achieved by increasing the ionic strength of the solution by the addition of sodium acetate and an alcohol, and then by harvesting the resulting precipitate by centrifugation. In the case of the use of a mini-column-based extraction technique, DNA is selectively eluted off the column with the use of a low-salt, high-pH buffer. The current slide illustrates two solution-based methods for DNA extraction. The first is the phenol-chloroform-based method, where samples are mixed with an equal volume of phenol-chloroform mixture. The proteins and the lipids partition into the organic phase, whereas the nucleic acids partition into the aqueous phase. The DNA can then be recovered by precipitation on the addition of sodium acetate and an alcohol. The second method illustrated is the CTAB proteinase K method, which employs a differential solubility approach for isolation of DNA and removing contaminants. DNA is soluble in CTAB in the presence of 0.7 molar salt, but precipitates if the concentration drops below 0.5 molar salt. The DNA can therefore be selectively isolated from the polysaccharide and proteins and further purified before being employed in any downstream applications. This slide illustrates the steps involved in the isolation of DNA using the CTAB proteinase K method, which was first described by Murray and Thompson in a 1980 nucleic acid research paper. The sample is first mixed with the CTAB extraction buffer and proteinase K, and then incubated overnight at an elevated temperature. The disrupted sample is then mixed with an equal volume of chloroform, after which the aqueous and organic phases are separated with the use of centrifugation. The DNA migrates into the aqueous phase, and the lipid proteins migrate into the organic phase. The aqueous phase is retained and mixed with an equal volume of CTAB precipitation solution before being centrifuged to collect the precipitated DNA. The pellet is then resuspended in a high salt buffer, re-extracted with chloroform, and the aqueous and organic phase is separated with the use of centrifugation. The aqueous phase is retained and mixed with an alcohol, and the DNA precipitate harvested by centrifugation as before. As an alternative to in-house DNA extraction approaches such as the Murray and Thompson CTAB proteinase K method, many labs now prefer to employ commercial DNA extraction kits. These kits commonly employ spin columns to isolate nucleic acids. The columns contain ion exchange materials that selectively bind nucleic acids, the process of which is dependent on salt concentrations and other factors. The process is comparably easier and faster to implement than conventional methods. However, troubleshooting can be problematic. This slide illustrates the typical workflow that would be followed when using commercial column-based DNA extraction and purification methods. Sample disruption is usually similar to in-house methods and involves the use of detergents such as CTAB or SDS, and enzymes such as proteinase K and chemical agents such as chaotropic salts. Following disruption, the DNA is bound to the matrix of the column, which can then be subjected to a number of wash steps to remove any impurities. These washes are normally comprised of a high percentage of ethanol or propanol and with a moderate salt concentration. And finally, the DNA is selectively eluted from the column using a buffer which has a low salt concentration but a high pH. The following series of slides will describe some of the methods employed for the quantitation and quality control of the extracted DNA. As an overview to the content of these slides, we will talk a little about why we need to characterise the extracted sample, describe some common DNA quantitation methods including ultraviolet spectrophotometry, fluorimetry and capillary or gel electrophoresis, how we evaluate the purity of a sample, usually through the use of ultraviolet spectrophotometry, how we can look at DNA degradation or fragmentation, otherwise known as integrity, by using gel electrophoresis, and how we can look for the presence of matrix interference. The decision tree presented here illustrates the basic steps that should be followed when performing a DNA quality control for DNA extracted from a sample. The quantity of DNA present is normally determined by measuring the absorbance of a DNA solution at 260 nanometers, or by measuring the level of fluorescence generated when it is bound by a special fluorescent DNA binding dye. The purity of a solution of DNA is normally determined by measuring the ratio of absorbance of ultraviolet light it produces at 260 nanometers and 280 nanometers and 260 nanometers and 230 nanometers. The integrity of the DNA can be determined by running it out on an electrophoretic gel, and the amplifiability of the DNA can be determined by using it to perform quantitative PCR using a serial dilution of the extract. Quantification of the extracted DNA can be determined using a number of different approaches, including ultraviolet spectrophotometry, fluorescence, that's the use of DNA binding dyes, for example picogreen, cybergreen or ethidium bromide, electrophoresis, or in a few special cases, real-time PCR. DNA quantification using the measurement of absorbance or optical density, as it is sometimes known, is the commonest method employed in DNA quantification. It's simple to perform, provides a low sample-to-sample variation, and utilises the Beer-Lambert law, which states that for double-stranded DNA in solution, an optical density of 1 corresponds to a concentration of 50 micrograms per ml. However, there are potential issues with the method. For instance, single-stranded DNA and free nucleotides cannot be distinguished from double-stranded DNA, and which can lead to falsely high quantification readings. In addition, inaccuracies can occur if the DNA is not homogeneously resuspended. Examples of instruments commonly employed for this type of quantitation include the Nanodrop 1000 from Thermo Fisher Scientific and the GE Healthcare's GeneQuant 1300. An alternative method for the quantitation of DNA employs using the measurement of fluorescence. This measures the fluorescent intensity of dyes that bind to double-stranded nucleic acids and selectively fluoresce when bound, for example picogreen. The method is particularly useful where the concentration is too low to be accurately assessed using ultraviolet spectrophotometry, and which broadly equates to a concentration of DNA less than 2 nanograms per microliter. Electrophoretic analysis can also be used to quantitate the DNA concentration of an extracted sample. In such cases, a sample of the DNA is run out on an agarose electrophoretic gel and detected with the use of a DNA binding dye. The technique has the added advantage in that it can give an indication of the level of DNA integrity present in an extracted sample. The quality of extracted DNA can be determined using a number of different approaches. For DNA purity, ultraviolet spectrophotometry is primarily used, and measurements of absorbance ratios at 260 and 280 nanometers and 260 and 230 nanometers are made. The integrity of the DNA is usually determined with the use of electrophoretic analysis, and matrix interference is usually determined by the detection of the presence of PCR inhibitors. The purity of DNA is conventionally determined using ultraviolet spectrophotometry. Critical measurement parameters include the absorbance ratios at 260 nanometers and 280 nanometers, and at 260 nanometers and 230 nanometers. Absorbance at 280 nanometers is indicative of the presence of proteins, whereas the absorbance at 230 nanometers is indicative of the presence of organic compounds or chaotropic salts, and which principally result from carryover of extraction reagents. DNA integrity is normally determined electrophoretically and using either conventional agarose gel-based electrophoresis or alternatively a capillary electrophoretic device such as the Agilent 2100 bioanalyzer. For the agarose gel electrophoresis system, when degradation is present, the high molecular weight top band visibly expands down the gel, producing a progressive smear or comet tail as the proportion of smaller fragments increases. In the case of the Agilent 2100 bioanalyzer, the electrophoretic fragment profiles show a visible increase in the distribution of fragment sizes, together with a shift of the distribution of the mean to the left, indicating a predominance of smaller fragments. The presence of matrix interference is normally determined by the detection of PCR inhibition. Numerous compounds can interfere with the TAC or related polymerases, and this may result in false negative results, for example, when no PCR product is generated even though the template is known to be present. Approaches to determine the presence of PCR inhibition include the use of an internal PCR control, for example, a spike in target for which a product should be generated, or use of a standard curve and evaluating this for linearity and PCR efficiency, or performing a serial dilution of the extract to see if inhibition is removed. For the purposes of providing guidance on quality control measures, we advocate the following: that the absorbance 260-280 ratio should be as close to a value of 1.8 as possible. Values appreciably lower may indicate the presence of protein, residual phenolic compounds, and other contaminants. That the absorbance 260-230 ratio should ideally be in the range of 2 to 2.2. If the ratio is appreciably lower than expected, it may indicate the presence of contaminants that absorb light at the 230 nanometer region of the spectrum. Gel-based analysis should result in a broad, brightly stained band positioned below the well. Smears or comet tails are indicative of degradation and/or fragmentation. Similarly, offset bioanalyzer profiles to the left are indicative of sample degradation and/or fragmentation. You should observe no impact of the template on PCR assay performance, and you should achieve the expected PCR efficiency and detect your product and/or internal control. This slide shows a decision tree which is applicable to the quality control of extracted DNA. Critical measurements in the decision process include that the optical density at 260 nanometers should be greater than 0.1. Values below this are indicative of low DNA recovery and re-extraction should be considered. Secondly, that the absorbance 260-230 ratio should be greater than 1.5. Values below this are indicative of the presence of a number of contaminants, and additional purification should be considered if possible. Thirdly, that the absorbance 260-280 ratio should be between 1.8 and 2. Values in excess of this are indicative of protein or phenol contamination, and again, additional purification steps should be considered. And finally, that no smears or comet tails are visible with agarose gel electrophoresis, as this is indicative of degradation and fragmentation, and DNA re-extraction should be considered. The following slides provide a summary of this section of the e-seminar on the general principles of DNA extraction and sample characterisation. We have looked at food matrices and seen that they provide a challenge for optimised DNA extraction. That sample preparation, including homogenisation and representative sampling, are key to achieving a successful outcome to the extraction process. That the objective of any DNA extraction procedure is to isolate, concentrate and purify DNA, and successful DNA extraction is key to any molecular test. We have covered the fact that there are a number of different methods for the extraction of DNA, with a range of commercial kits and in-house methods being available, and that these can be solution-based, solid phase-based, or a combination of the two. We have provided a general overview to the DNA extraction procedure, including a brief description of sample disruption, release of DNA and removal of cellular components, isolation of the DNA either through its binding to an inert matrix or by using precipitation techniques, washing the DNA to remove impurities, and finally we briefly covered resuspension or elution of the purified DNA. And finally we talked about characterisation of the extracted DNA sample, including approaches to evaluate both its quality and its yield. In this section of the e-seminar, we will cover DNA extraction methodologies applicable to the foods area. During the following series of slides, we will give an overview of food-specific DNA extraction methodologies, including those specified for use in the UK and other EU official control laboratories. We will look at some common approaches employed in the extraction of DNA from food, including examples of commercial kits and in-house techniques, and give some detail on specific approaches such as the CTAB technique. We will also provide some guidance on DNA extraction troubleshooting, as well as suggestions for performing quality control on the extracted DNA, including the provision of a number of decision trees and working recommendations. Finally, we will look at matrix interference, including its identification through the detection of PCR inhibition, and provide some recommendations as how to proceed if its presence is identified. In the following few slides, we will focus on those DNA extraction methods commonly used with foods. In the following series of slides, we will provide an overview of those food-specific DNA extraction methodologies employed by both UK and other EU official control laboratories, and look at some of the common approaches, including examples of commercial kits and in-house techniques. If we were to consider those DNA extraction methods commonly used with foods, then a review of the relevant scientific literature reveals that a wide variety of in-house and commercial kits are available for the purpose. These are both universal and matrix specific in their application, but overall limited practical guidance is available to recommend which should be selected for any particular purpose. For illustrative purposes, examples of both in-house methods and those available as commercial kits are listed on this slide. This slide provides examples of those nucleic acid extraction methods that have been cited in FSA and Defra food project reports and serves to illustrate the broad range of methods that have been employed. However, it can be seen that the use of a CTAB-based extraction buffer in conjunction with the use of a solid phase purification step has frequently been employed in several of the projects. This slide provides details of those DNA extraction techniques that have been recommended in EURL guidance documents for processing different samples. As seen with the previous slide, there are a number of DNA extraction methods that are recommended. However, of note is the fact that the use of the Wizard magnetic DNA purification system is a binding requirement of the SOP for extracting DNA from animal feeds. In the preceding couple of slides, we've given examples of DNA extraction methodologies used within the food sector, including those cited in FSA and Defra project reports, and no cited in EURL guidance documents. We have seen that there are a variety of in-house and kit-based approaches in use, and that many of these employ a CTAB-based approach. That these include extraction from raw and processed food types. And finally, we've mentioned that at least one of the EURL methods is included in a binding SOP. The following slide provides guidance on selecting an appropriate DNA extraction approach. We've seen that there are a variety of approaches in use, but we've selected the most widely used in the food sector, and for which we've provided details on their applicability to food matrices, their key features, performance details, and basic costings. This slide provides guidance on the selection of appropriate DNA extraction techniques for use with a variety of food-related products. The list is not exhaustive, but provides details of those methods currently in use within the foods area. This includes methods suitable for use with animal and plant-derived products, as well as fungi, fruit juice, oils and lipids, as well as chocolate. The following couple of slides serve to provide some more detail on those DNA extraction techniques which employ CTAB. Development of the CTAB-based DNA extraction technique is credited to Murray and Thompson in 1980. It's applicable to the extraction of DNA from plants and plant-derived materials, but is particularly suited to where high levels of polysaccharide and polyphenolic compounds may be expected. Of note is the fact that it's also useful with many other matrices, including meat and meat-based products. It relies on the disruption of a sample at an elevated temperature and in the presence of CTAB, but enzymes including proteinase K and RNase A are frequently included. Chloroform is used to separate the nucleic acids from the CTAB and polysaccharide protein complexes, and salt concentration is important to the removal of contaminants. This is because CTAB nucleic acid complexes precipitate below salt concentrations of 0.5 moles per litre, resulting in many contaminants being retained in solution and effectively removed. We see presented here a simplified workflow of the basic CTAB DNA extraction method developed by Murray and Thompson. The sample is mixed with a CTAB extraction buffer plus proteinase K and incubated at an elevated temperature to achieve sample disruption. The disrupted sample is then mixed with an equal volume of chloroform, followed by phase separation of the aqueous and organic layers using centrifugation. The aqueous phase is retained and mixed with the CTAB precipitation solution, which causes the DNA to precipitate. The precipitate is harvested by centrifugation and solubilized in a high salt buffer. The solution is extracted with chloroform, and the aqueous and organic phase is separated using centrifugation. The aqueous phase is retained and mixed with ethanol to precipitate the DNA, and the DNA is harvested by centrifugation and the pellet washed with 70% ethanol to remove excess salt and residual CTAB. The pure DNA is then air dried and then resuspended in an appropriate buffer. The procedure depicted here illustrates a modification of the basic CTAB procedure, and which employs the use of a DNA binding column to purify the extracted DNA. With this approach, the DNA recovered from the first round of precipitation is bound to a DNA column, washed to remove contaminants and eluted. Use of the columns avoids the need for a second round of chloroform extraction and reduces the risk of carryover of reagents or co-purifying contaminants. As a guide to performing CTAB DNA extraction procedures, we make the following recommendations: that you store all CTAB containing buffers above 15 degrees centigrade to avoid the CTAB precipitating out of solution, that the optimal tissue to buffer ratio requires empirical evaluation, but as a rough guide allow 1 ml of buffer to every 30 to 100 milligrams dry weight material. Perform overnight extractions at 56 degrees centigrade and cool the extraction buffer to room temperature prior to adding chloroform. phase separation is best achieved with the use of centrifugation, and we recommend performing a re-extraction of the final aqueous phase with chloroform prior to the final DNA precipitation step. Avoid using high centrifugal forces, as these can result in CTAB carryover, and we recommend washing the final DNA pellet with 70% ethanol to remove excess CTAB, as CTAB is soluble in ethanol. The following slides will provide general guidance on troubleshooting DNA extractions. As an overview to this section, we will provide some general guidance for problems encountered with DNA extraction in general, provide guidance on dealing with problems associated with column-based approaches, and provide recommendations for extracting DNA from different food matrices. Frequently encountered problems of DNA extraction procedures include low yields, the presence of sheared or fragmented DNA, as well as low absorbance to 60 nanometers to 80 nanometers and absorbance to 60 nanometers to 30 nanometers ratios. In the case of low yield, incorrect sample storage, too much starting material, or incomplete sample disruption are frequently the cause. It's worth remembering that the yield of DNA you can expect is dependent upon the type, age, and storage conditions employed for the sample you are extracting DNA from. Therefore, it's always worth checking the method of collection that has been employed and also the conditions of storage. Where too much starting material is suspected, try initially reducing the amount of material you're using, or cut the sample into smaller pieces to maximize the surface area to buffer ratio, or try using a different homogenisation method. Where incomplete sample disruption is evident, in that you still have large clumps of sample remaining after incubation, try including a proteinase K digestion step, or if this is already being employed, increase the amount of proteinase K you are adding and increase the duration of the incubation. Where sheared DNA is evident, this is frequently the result of a sample being subjected to multiple freeze-thaw cycles, or just that the samples are too old. Try to avoid subjecting samples to repeated freeze-thaw cycles, as this will result in the release of nucleases that will fragment your DNA. The physical action of ice crystal formation in your sample can also shear the DNA. Therefore, where possible, aliquot your sample so that you only thaw each aliquot once. Also, try not to use old samples, as the DNA in these is frequently degraded as a result of the action of a number of mechanisms. Where the absorbance to 60 nanometers to 80 nanometers ratio is less than 1.8, protein contamination is frequently the cause, although it can be indicative of the carryover of phenolic compounds resulting from your extraction reagents. Try using a proteinase K digestion step if you suspect the cause is protein carryover, or try performing an additional chloroform extraction step, as this will aid removal of both proteins and phenolic compounds. You will then need to perform an ethanol precipitation or column purification step, however, in order to remove the residual chloroform. Where the absorbance to 60 nanometers to 30 nanometers ratio is below 1.5, this is usually indicative of extraction reagent carryover. Therefore, include the use of an additional solvent extraction step using chloroform to remove these contaminants, followed by an ethanol precipitation of the DNA, washing of the resulting pellet with 70% ethanol before air drying the pellet and resuspending the DNA in a suitable buffer. This slide provides some general guidance for dealing with problems associated with column-based DNA extraction methods. Common problems include low yields, clogged columns, DNA does not perform in downstream applications, and a white precipitate appears in the column binding buffer. For low yields, possible causes include the insufficient mixing of sample with binding buffer and ethanol, that the DNA has not been eluted efficiently, that the column binding buffer has been incorrectly prepared, or that an incorrect eluent has been used to elute the DNA from the column. Where insufficient mixing is suspected, try repeating the whole process with a fresh sample, but ensure that adequate mixing of buffers prior to use and sample buffer mixes is performed. As a rule of thumb, we advocate mixing by vortexing for at least 15 seconds for each step in a protocol where you are required to mix solutions. Where inefficient elution is suspected, firstly check that all the buffers have been prepared and mixed correctly. Then repeat the extraction process with a fresh sample, but increase the volume of elution buffer you've previously employed, or perform multiple elutions and pool the aliquots. It's worth noting that many kits recommend or provide specific elution buffers for the purpose, so it's worth confirming with the protocol as to what to employ. One option to increase the yield can be to warm the elution buffer to 70 degrees C before use, and on application to allow the column to stand for 1 minute before recovering the DNA by the normal centrifugation method. In the case of clogged columns, the usual cause is starting with too much material, or that insufficient sample disruption has been achieved. This can sometimes be remedied by increasing the G-force of the centrifugation, or preferentially by extending the duration of the centrifugation step to avoid damaging the integrity of the column. When downstream applications fail to perform, as would be expected, it's likely that reagent carryover has occurred, and this is usually salt or ethanol. To avoid salt carryover, ensure that all the buffers are at standard room temperature or at the temperature required by the protocol prior to use. To avoid ethanol carryover, it's essential to avoid contact of the column with any of the flow-through, especially during any centrifugation steps, and it's frequently recommended that a dry spin be performed prior to elution to ensure all residual ethanol has been removed. Finally, where a white precipitate is observed in the column binding buffer, this may be the result of protracted storage at suboptimal temperatures. The problem can usually be remedied by warming the buffer to 56 degrees centigrade until the precipitate redissolves. It's often possible to adjust a DNA extraction method in order to allow its use with different matrices than it was initially designed for. For example, matrices with a high fat content can be accommodated by the inclusion of a solvent extraction step, whereas samples with a high starch content can be dealt with by the inclusion of an enzymatic alpha amylase digestion step. For samples with high polyphenolic or tannin content, the use of a CTAB extraction approach in conjunction with an ion exchange column step is frequently recommended. Samples with high protein contents can be addressed by the inclusion of a proteinase K treatment, whereas samples with a high heme content can be dealt with by using the CTAB-based approach in conjunction with an ion exchange column. In the following few slides, we will provide guidance on how to characterise extracted DNA samples, specifically how to evaluate yield, purity and quality. As an overview to the content of the following slides, we will provide some sample quality control decision trees to help guide you through the quality control process. We will provide some general recommendations on how to implement a quality control assessment, and we will talk a little about the topic of matrix interference, how to determine the presence using PCR, and recommendations for dealing with the problem. This slide is the first of two slides which provide an example of a decision tree for a spectrophotometric and electrophoretic gel-based sample quality control system for use with extracted DNA. Initially, the absorbance at 260 nanometers is evaluated. For values less than 0.1, low recovery of DNA is indicated, and the sample should either be re-extracted, concentrated using an ethanol precipitation, and resuspension in a minimal volume, or concentration by passing the solution through a commercial microfiltration concentration device. If a value in excess of 0.1 is obtained, then the next step is to evaluate the absorbance to 60-80 ratio, with an optimal value for this being in excess of 1.8. Values less than this are indicative of protein and/or phenol contamination. Corrective actions that can be implemented include repurification of the sample or re-extraction with the inclusion of a proteinase K step in the case of a suspected protein contamination. Provided the criteria for the absorbance to 60-80 ratio are satisfied, then an aliquot of the DNA should be subjected to electrophoretic analysis. The lack of a smear and the presence of a bright high molecular weight band below the well are indicative of primarily intact DNA. If the sample is marked by the presence of a smear or comet tail, then the isolated DNA is fragmented and degraded, and re-extraction should be seriously considered. Provided all of the above quality criteria are satisfied, then the sample can be progressed to the next stage of the quality control process. The decision tree presented here follows on from the example shown on the previous slide. Provided the extracted sample has passed all of the quality criteria specified on the previous slide, then the next step is to evaluate the absorbance ratio at 260 nanometers and 230 nanometers. Values obtained for this should be in excess of 1.5, and values below this are indicative of the presence of phenolate ions, salts and polysaccharides. Corrective actions that can be undertaken in the case of suspected phenolate ion contamination include either re-extraction with a fresh sample or chloroform extraction of the original DNA solution, followed by ethanol precipitation of the aqueous phase in order to recover the purified DNA, and resuspension of the resulting DNA pellet in an appropriate buffer. Where contamination with salts is suspected, the DNA can either be purified by ethanol precipitation to recover the purified DNA, and resuspension of the resulting DNA pellet in an appropriate buffer, or the salts can be selectively removed with use of a commercial cleanup column. Once the absorbance to 60-30 ratio criteria have been met, the absorbance at 330 nanometers should also be measured, and for which values greater than 0 are indicative of particulate contamination and which can originate from a number of sources. However, this is usually remedied with the use of a short, high-G centrifugation step to pellet the particles and transfer the DNA solution to a clean tube. Once all of the quality criteria have been met, the sample can then be considered suitable for further analytical procedures. This third quality control decision tree is based on a PCR evaluation of the sample to gauge its applicability for use with this type of application. The first step in the process is an assessment of the PCR negative control, for which no amplification should be detected. Provided this requirement is satisfied, then the presence of amplicon in the extracted sample is checked for, and if present, if it is of the correct and expected size. Provided these criteria are satisfied and no amplification has been detected in the PCR negative control, then the analysis can be proceeded with. A number of alternative and remedial actions have been included at each of the breakpoints. In terms of some general suggestions for implementing a quality control procedure, we advocate the following if the sample fails quality control. Where practical, the DNA should be re-extracted using a fresh sample, and the appropriate quality control procedure repeated. Alternatively, and if re-extraction is not possible, we suggest trying to remove some of the contaminants and any small DNA fragments using either in-house methods such as ethanol precipitation or by using one of the many commercial cleanup columns available to remove contaminants and concentrate the DNA. In the following few slides, we will cover the topic of matrix interference. As an overview to these slides, we are going to look at PCR inhibition as a means for detecting the presence of matrix interference. We will mention some commonly found PCR inhibitors, both food-based and extraction reagent-based. We will look at a mechanism to detect PCR inhibition, and then we will provide some guidance on how to employ PCR inhibition testing. With respect to PCR inhibition, a large number of compounds are capable of interfering with TAC or related polymerases. The presence of PCR inhibition can be indicated by faint and or sporadic amplification, poor CV values for replicates, and late CQ values for quantitative PCR assays. This may result in false negative results, for example, no PCR product being produced even though the template is present, or even the under-quantification of the target. This slide lists some of the common PCR inhibitors which are present in both food-based products and extraction-based reagents. Food-based inhibitors include complex polysaccharides, which are found in plant materials, collagen, which is found in animal connective tissue, the heme from blood, humic acids that are present in soil and plant materials, proteinases, which are present in milk, as well as immunoglobulins, which are present in animal blood. In terms of extraction reagent-based PCR inhibitors, SDS is by far the most inhibitory, and at a concentration of 0.0005% mass for mass. Phenol is inhibitory at around 0.2%, and most alcohols have an effect from 0.2% as well. Two approaches can be employed to determine the presence of PCR inhibition. Firstly, the use of an internal PCR control, or IPC, which is used both in the presence of the DNA extract and in the absence of the DNA extract, or alternatively by employing a linear dilution series of the extracted DNA to examine, in the case of endpoint PCR applications, the PCR amplicon band intensity and the linearity of the data associated with this. And in the case of quantitative real-time PCR, by looking at the metrics describing the resulting standard curve, as well as the values for the PCR efficiency of the assay that was employed for the analysis. Detection of PCR inhibition using an internal PCR control, or IPC, is performed in the following way. Your internal PCR control normally takes the form of an IPC-specific target, along with IPC-specific primers and probe. You perform two reactions. In reaction 1, you have the IPC assay plus water, and in the second reaction, you have the IPC assay plus a dilution of your extracted DNA. You then examine the results obtained, and when considering endpoint PCR, you need to look for the absence of a PCR product or reduction in band intensity when running the product out on an agarose gel. When considering real-time PCR, you look for an increase in the resulting CQ. The detection of PCR inhibition can also be achieved by using a linear dilution series or standard curve method when utilising a real-time PCR approach. A 10-fold series dilution of your target DNA should result in a CQ difference of 3.3 cycles between successive dilutions. Plotting log 10 transform values for target copy number against CQ will generate a curve for which a negative slope gradient should be obtained. The PCR efficiency of the assay can then be calculated in percentage terms by multiplying 100 by 10 to the power of -1 over the slope of the line, -1. A PCR efficiency of 100 equates to a slope of the line of minus 3.3 and relates to a twofold change in CQ value. PCR efficiency should be between 90 and 100%, although values of 100 plus or minus 15% are frequently referred to in the scientific literature. The slope of the line should be between minus 3.6 and minus 3.3. The R-squared value for the line of best fit, and which expresses a measure of linearity of your data, should be between 0.95 and 1.0. Lack of linearity and poor PCR efficiency are a good indication of the presence of PCR inhibition. In the example illustrated here, the slope of the line has been determined to be minus 3.386, and which corresponds to a PCR efficiency of 99.5%. The impact of the slope of the line on PCR efficiency is illustrated in the box at the bottom right hand side of the slide. An optimal slope of minus 3.32 equates to a PCR efficiency of 100%, whereas a slope of minus 4 only equates to an efficiency of 78%. In terms of when to test for matrix interference, we would advocate the following: that you test when using a non-validated purification protocol, or when a validated protocol is used but the sample matrix has been changed, if the amount of input material is varied from that stated in the validated protocol, if a new operator has performed the purification, or when the protocol is customized, for example an additional wash step is added, or finally when the experimental samples may be heterogeneous in nature, such as with environmental samples or processed foods. The following series of slides will summarise the information provided in this section of the e-seminar. Firstly, we have provided examples of methodologies employed within the food sector and that are detailed in FSA, Defra and EURL publications. We've described a variety of in-house and kit-based approaches and shown that they frequently employ CTAB. We've provided guidance on the performing of extractions and tips for general troubleshooting. We've described the use of spectrophotometric methods to examine extraction sample purity, including the absorbance 260:80 ratio, for which the critical value should be in excess of 1.8, the absorbance 260:30 ratio, for which the value obtained should be in excess of 1.5, and the absorbance at 330 nanometers, for which a value approaching 0 is considered optimal. We have explained that DNA integrity can be evaluated using gel electrophoresis, where the absence of visual smears and comet tails is desirable, or with use of an adjuvant bioanalyzer, where a high mean fragment size and a minimal fragment size distribution are indicative of a sample with acceptable integrity. That the amplifiability of extracted DNA can be tested for by use of an internal PCR control, or by evaluating assay performance with use of a serial dilution series of extracted DNA, and looking for R-squared values for the resulting data in excess of 0.99 and PCR efficiencies in excess of 90%. And finally, we strongly recommend that you employ some form of quality control for your extracted DNA before employing it in any additional molecular analysis. The KT event presenters and food analysis team at LGC comprise of Malcolm Burns, the Principal Scientist for Food Analysis at LGC, Gavin Nixon, Claire Bushell, and myself, Tim Wilkes. And finally, I'd like to thank you for your attention.